It is my pleasure to introduce Anand Tambur, who is here today from Northwestern University virtually. She's going to be talking about HLA, and we are really excited to have her. Things I did not know about Anand. She was a dentist in a previous life. That is something I just found out, and that's fantastic. But she runs the HLA lab as well as the biorepository at Northwestern University in their transplant center, and has had extensive leadership roles in the American Society of Histocompatibility, as well as significant involvement with the American Society of Transplantation. And her knowledge in this area is unparalleled, and we're very fortunate to have her virtually today talk with us and update us on some of the work that she's been doing. So with that, I will turn it over to you.
Fantastic. Thank you, Lara. And yes, dentist is kind of like dentistry, I should say, a dark moment in my life. I got my diploma and my license put in a drawer and realized that this is not something I want to ever do in my life again. But yes, I've done that. So today we're going to talk a little bit about the complexity of HLA-DQ and its significance in solid organ transplantation. And I want to share this with you. This is actually a painting that I commissioned looking at an HLA-DQ molecule. I have a 3D structure of it. So you look at the alpha chain, the beta chain, the peptide in between, and this is just to kind of give you a sense of the complexity of how HLA-DQ molecule is involved. I'm especially focusing in solid organ transplantation, but obviously in autoimmunity as well. So with that very colorful figure, let's start. We are going to talk about the involvement of HLA-DQ in solid organ transplantation, but HLA-DQ, like all other HLA molecules, actually have a significantly more important function in physiology and in health. Both molecules are there in order to present peptides to the T cell arm of the immune response. So we have the class 1 molecules presenting peptides to the T cell receptor, and class 2 antigens, presenting peptides to the CD4 arm of the response. And while there are some differences, for example, class 1 molecules will usually present intracellular peptides. It might be viruses. Oftentimes it's just self-proteins from housekeeping genes, etc. And they have a particular antigen processing pathway that will come to the cell surface versus class 2 that usually present extracellular molecules that are brought into the cell and being digested, processed, and brought into the cell surface. But as you can see, the structure of those molecules, and that's a little bit better example, is very, very similar. 4 extracellular domains for class 1, A, B, and C that are encoded within chromosome 6. We have a gene that encodes for the 3 polymorphic extracellular domain of the molecule, and then it's helped by a beta-2 microglobulin to form this 4-domain structure with a peptide presented here. Class 2 molecules are also encoded in chromosome 6, also have 4 extracellular domains, but those are encoded by 2 genes, 2 separate genes within chromosome 6. The A and the B gene that we have both for all DRDQ and DP loci, where the A gene encodes the alpha chain with 2 domains, as you can see here, and then the B gene encodes the beta 1 and the beta 2 domains of the beta chain. The important thing is, though, that the full structure is those 4 extracellular domains. And for those of you that deal with transplantation and talk about HLA antibodies, and really from now on, we're going to talk about the antibodies, not so much the DQ molecule per se. You can hear people talking about DQ alpha antibodies, DQ beta antibodies, and I just want to start with that. That there are no single alpha chain or single beta chain that is on the cell surface. We're always talking about a heterodimer, the 4 extracellular domains for class 2, the same that we have for class 1. So that's just kind of like laying the foundation to what we will be talking about moving forward. Another piece of information just to kind of get us going is how we test for HLA antibodies in the lab. This is an example of a worksheet for a single antigen bead assay or SAB assay, as we usually call it, looking at class 2. And you can see just in, random numbers given by the vendor for a bead ID and the vendor's name for this. And you can see the MFIs here, but really what I want you to focus on is what we refer to as the serological typing. This is the old serologic nomenclature. And then the molecular typing that is listed a little bit differently. You have the locus and an asterisk and the allele name as you're looking at. But just as an example, if we're looking at DR7, we're looking at molecularly DRB10701. Now you say, hey, you just said that you have to have an alpha chain and a beta chain. Why are you talking just about DRB gene here? What happens with the alpha? And that's a hidden secret of the HLA people. The alpha chain of HLA-DR is non-polymorphic. There is no polymorphism, none whatsoever. I'll take it back. There is one amino acid difference or one nucleic acid difference that does not contribute to protein differences. So we just don't talk about the alpha chain when we talk about DR, but there is an alpha chain. It's consistent among all the different alleles. So when you look at DR, you'll hear DRB1 with the specific alleles. When we move to DQ and DP, alpha chain is polymorphic. The DQ alpha is polymorphic, the DQ beta is polymorphic. So this is just looking at the same continuation of that same page, but now we're focusing on HLA-DQ. And if you look at the serological typing, you can see that there are multiple DQ2s, multiple DQ4s, et cetera, et cetera. But if you look at the molecular typing side of it, you'll see that at least one of the chain is going to be different. So here we have all DQB02 chains, and O201 and O202 are really very similar, but different alpha chains or different DQA1 genes. And all of them serologically will be called DQ2. Molecularly, they are different. And what I hope I'll convince you today is that those differences are going to be really important as we're moving ahead. So one of the things that I was interested in for many, many years was what does the antibody recognize? And this is coming from a paper that was out about 8-9 years ago. And we wanted to focus on HLA-DQ epitopes. That's about the time where HLA Matchmaker, the software that was put forth by René Duquesnoy, became very commonly used, and we wanted to see what we can learn from this. So we had a study where we looked at patients that were 0% PRA pre-transplantation. We had multiple samples of them, so that was as good as we can to say they were non-sensitized prior to transplantation. They got an organ, they lost it, and developed donor-specific HLA-DQ antibodies. For inclusion criteria, we must have DNA from the patient to be able to high resolution type these alleles. And we refer to the patient as the antibody producer. We must have donor DNA to be able to high resolution type, again, the same alleles. We wanted to look at the DRB1 to look at the impact, but also that is really important to know how to pair the alpha and the beta together. So we refer to the donor as the immunizer. And as I said, we used HLA Matchmaker in order to identify putative applets. So what you're seeing here is a different way of looking at the single antigen bead assay. You can have the DQA, DQB genes here. You have the MFI values. You have the number of applet mismatches between the antibody producer and the immunizer and the antibody that the patient had formed. You can see that many of them were negative, but quite a few of them were positive. And the software allows you to look at the identity of those different applets. So the arrow here represents the donor's typing, the immunizer. The patient was 0% PRA. So really, when the patient developed antibodies to this particular DQ type, that antibody recognized a lot of other DQ specificities. This is part of the concepts behind HLA matchmaker. Again, not interested in the number of applet mismatches, but the reasoning was, well, if all of those antibodies were generated just by recognizing this particular antibody, they must have something in common. And indeed, you can see here that all of them have an applet at position 77 referred to as DR, and we thought at the time pretty naively that that is the core applet, that this is what's making the epitope of the specific donor-specific DQ antibodies. One can use a software from the NIH, it's called CN3D, looking at the alpha chain. So this is the the donor DQ molecule, the alpha chain here in pink, the beta chain in blue. This is the peptide that is presented normally within every DQ, every class 1, every HLA molecule. And this is position 77. Right, so you can see it in the beta chain. And one of the beautiful things that we can do with the CN3D, we can take that core and ask the software to show us whatever radius around that particular applet. Again, at the time, the notion was, unfortunately today, by many as well, that the applet is the center of the epitope. The epitope has a radius of 15 angstroms. So this is what you see here in yellow. And when you excise that portion of the HLA molecule and just remove the yellow color and look at what is the footprint of the antibody. So again, I'm not sure it needs to be a radius of 15 angstrom, but we are somewhere there and about. Then here's our applets at position 77, but the antibody would recognize this complete footprint. So it will include portions of the alpha chain, actually will include a portion of the peptides, and definitely a portion of the beta chain. I'll share quickly another example. So here are the antibody producer, here is the immunizer. You can see all the positive and the negative responses here. This is the immunizer HLA-DQ A and B combination. You can see the applets on the beta chain, the applets on the alpha chain, and the only set of applets that can explain why a non-sensitized patient pre-transplant will suddenly show all of those antibodies after being exposed to the immunizers are two adjacent applets at position 60 and position 64. So again, going through the same exercise as I showed you before, here is position 60 and 64 just adjacent to it. We'll go through putting a radius of 15 angstrom around it, removing this yellow color, and now we see an applet that is at position on the alpha chain, but still the footprint of the epitope will contain portions of the alpha chain peptide as well as the beta chain. So this study included 40 patients. I know it's not a lot, but I feel that this is a very rigorously curated study that we felt that we can assign the antibody responses post-transplantation to that one event of sensitization for those patients and have the sensitizing DQ typing available from the donor. What we found is that over 80% of the applets were indeed located on the DQ beta chain. That makes a lot of sense because the serologic nomenclature that we have that is based on antibody recognition of the antigen in serological assays, the beginning of the HLA days, 80% of them were located to the beta chain. but actually all of them, other than one case, the epitope footprint, right? So if we're looking at roughly those 15 angstrom, included portions of the alpha and the beta chain together. And from that point on, it was really clear for us that we must always look at the alpha beta combinations as one unit and always refer to it as such. It does have clinical implications. I'll share a couple of examples here just to demonstrate this. We are looking at a single antigen bead assay of one patient. I'm sure you're used to seeing those. You have your MFI values on the left, on the y-axis, and the HLA class 2 specificities here in the bottom. And I've highlighted two different beads specifically, and you can see that both of them are considered DQ2, right? The terminology goes by the DQ beta chain, but they have different alpha chains and they have similar MFIs. What I don't show you right now is other portions of the same single antigen bead assays. And here we have additional HLA-DQ2 alleles, and those are completely negative. So the question is, if we have a donor, do we have a DQ2 DSA or not, right? We need to know the alpha beta information. And even more tricky is if we need to list unacceptable antigens for these patients, are we going to list DQ2 as unacceptable or not? And we don't have currently the ability to make the distinction between those different alleles when we list unacceptable antigens in the unit. Why is it so important? We have looked at about 4,000 haplotypes from our own population. So the percentages may differ a little bit around the country, but those are haplotypes that we felt very comfortable that we can generate based on family studies. And you can see that the DQA 0501, 0201 here in blue comprises a little less than half of the DQ2 positive haplotypes versus the other very frequent allele 0201, 0202 in this case is down here is completely negative. So again, are we going to list this as unacceptable or not? Are we willing to accept positive cross matches by not listing this, or are we willing to miss a lot of potential donors that might be negative if we don't list this as unacceptable? And we've been debating this a lot and actually been dinged quite a lot for not listing some of those DQ2 positive patients because we didn't want to miss any potential opportunities. Another quick example, just to show you that it does not pertain just for DQ. So the molecular DQ-B0301 translates to DQ7 serologically, and you can see the DQ7 alleles in this case. The 0301 allele comprises about, or it's present in about 20% of the DQ7 positive individuals. On the other hand, the more frequent allele in this case, which is the 0501, 0301, is down here. We actually call it 05G. It's a combination of 0501, 0503, or 0505 for the alpha chain. It doesn't matter. All of them are completely negative. So if we're going to list 7 as unacceptable, for example, we're going to miss 2/3 of our donors if we don't have the high resolution typing of the donor and we're looking for DSAs. We may mistakenly call a DSA if our donor is actually this type. So it really has a lot of clinical implications. And the whole issue of listing unacceptable was something that we wanted to continue exploring. Unfortunately, with the SRTR database, as I told you, we cannot put that information in. Just to kind of calm the clinicians on the call, if you are looking for an offer in the middle of the night, I'm sure your HLA lab pulls the HLA attachment, so we can get that information in real time, but it is not captured in the SRTR as of now. The HLA attachments, I think, have been around for at least 4 or 5 years, so we can do good consults, but we cannot do good studies later on to look at those patients. So I resorted to rely on my friends in France, in Paris, and they had provided me DNA samples of about 2,500 recipients that were on the wait list during, I believe we've done the study in 2013. Since I got their DNA, I was able to high resolution those recipients for their DRB, DQA1 and DQB1. They also gave me DNA from a little over 1,200 consecutive effective donors. So they were used in transplantation during those years, and again, performed the same typing on them. And the important thing, I was able to then derive the frequency of the different DQ alpha combinations in this particular cohort. And then we use those frequencies to help us assign CPRA and look for potential compatible donors. And I think it was a really interesting exercise. You can see the composition of that cohort here. So we wanted to look at what might happen to the CPRA using the UNOS way of calculating things. If we convert the CPRA from where it was using the serological CPRA, the way that UNOS is using it, or converted to what we call the DQ allelic CPRA. So we kept A, B, C, DR the same way as it was, but replaced the DQ antibodies with the accurate alpha beta antibodies and compare it to the donor frequencies of the alpha beta calculations. You can see that CPRA is giving here in groups of 10 other than those that were greater than 95. And again, we're always really interested in those very highly sensitized patients. And you can see that all the serologic CPRAs would be here at our 0% decrease, of course, of the CPRA. And you can see how much decrease we can get in the CPRAs if we just call the DQ antibodies by their alpha beta combination and not take the whole antigen entity as a potential antibody, like the case that I showed you with the DQ2, like the case that I showed you with the DQ7. And we have a lot of patients that have allele level antibodies to DQ specificities, not comprising the whole antigen group. So again, if we'll focus here on the highly sensitized patients, there are quite a lot of patients, and some of them would lose significant amount of their CPRA. This is really important because we provide allocation points based on the CPRAs, right? So we can look at the reduction of the allocation points. This is again the UNOS scheme, whether the CPRA was calculated by the serology metric, which is what we're using currently, or what we proposed using the DQ allelic change to this formula. We just arbitrarily use MFIs of 1,000 in order to determine positivity. That can change, but and we actually looked at this. The projections were very, very similar. The numbers would have been a little bit different. And again, I'll kind of point to the patients that receive 100% CPRA points. So this is actually everyone from CPRA of 99.5 because we round up those numbers. Using the current system, they would have been 456 patients. But if we will correct the CPRA based on the DQ alleles, we'll get a 37% decrease of the numbers who will qualify to those 100 points. And I think this is really important because in my mind, patients that are at this point can get a lot of compatible donors, right? So they're getting a lot of priorities, while the patients that truly remains 100% CPRA are trying to get compatible donors, but they're not, they're in the same group with patients that can do significantly better. We also highlighted on this slide, excuse me, patients that were greater than 85% VPRA, virtual PRA. This is the system that was happening in France. And every patient with VPRA of greater than 85% will get additional points. It's not that very significant escalation on allocation points. So that was not as significant as we've seen for the American population. And of course, we can see a reverse effect to the CPRA on the effect of changing the DQ from serologic to molecular allelic alpha beta combination CPRAs. Now the number of potential compatible donors will be significantly higher. And again, pointing out those really highly sensitized patients, some of them can get a lot more compatible donors. So again, if we want to make sure that we're maintaining equity on the system, if we want to do the best for our highly sensitized patients, I think we really need to pay significantly more attention to what's happening with the DQ. And of course, it will affect the other patients that are on the wait list, even those that have relatively low CPRA points. We wanted to try and give it a little more kind of flavor on what will happen with the different blood groups. And you can see them listed here. I hope you don't see that part of on the screen. I don't know how to make it go away. I'm sorry. So it should be, I believe, A, B, B, and O. And this is the number of potential compatible donors that each of the patients in these blood group categories will get. Here you look at the median, for example, for the blood group O. And this is what will happen with the median if we're going to use the allelic alpha beta combination of the DQ typing. So again, we can help the patients that really need to get a little bit more help in order to find a compatible donor. We looked at the very highly sensitized patients in a very awkward way. We didn't have a way to simulate it better. We just decided to filter the patients to those that can have a maximum 20 potential donors. And again, this is the serologic one. So capping it here at 20 patients. And you can see what happens when we are moving to the allelic way of doing things. So these are the very highly sensitized patients. And again, some of them can get a lot more potential compatible donors. So in that case, we can see how DQ can affect the CPRA, it can affect the allocation. We also know that HLA-DQ is the most frequent de novo DSA. We know it's the most detrimental DSA. And I could quote a lot of different studies. I chose to bring, obviously, the one that was done in your center that really support that point. And when we have this in mind, and we're thinking about how we're doing matching or how we are considering matching in the US. We're looking at 0 ABDRs. DQ is not even mentioned in this. Not that we're giving a lot of priority points for matching, but DQ is totally not in the equation. So I think it would have made sense to include HLA-DQ as part of the matching algorithm. And just to, again, reiterate, unfortunately, we cannot get good data from the SRTR to do this. So we published just recently an opinion paper to talk about the importance of looking at what HLA antibodies do for patients who need to return to the waiting list, because we cannot have a big cohort that would look at the impact of ABMR. The data is not available in the SRTR, but this is looking at SRTR patients. This is a snapshot for somewhere in 2020. And you can see the distribution of CPRAs for patients awaiting primary transplants. The over 80% and over 98% are listed here, right? So patients awaiting their first transplant are not very highly sensitized. This is what happens when you're looking only at the patients that are coming for a repeat transplant. About half of them come back to the wait list with CPRAs greater than 80%, more than a quarter have a CPRA greater than 98%. And this is even more prominent when you're looking at the pediatric patients. So you can look at this a little bit different. We just grouped the CPRAs in those buckets. And so, for example, in this particular categories, in purple, you can see the percent of patients that have this particular CPRA when they're awaiting their primary transplant. So they're about 70% of the adult waitlist patients have CPRAs of greater than 98 and greater. And if you look at the pediatric patients, you see that 90% of the repeat pediatric waitlist will be composed of patients with 98%. And this is really heartbreaking, because those patients will need to get their second transplants and oftentimes a third transplant, and if they're lucky, maybe even more. But we're making them so very highly sensitized because we don't have any consideration for HLA matching in this regard. And we talk about, well, let's not consider HLA matching because it's going to disadvantage some population, and the finger usually is on the African American population. So let's look at what's happening when we're looking at the ethnic distribution of your CPRA for the primary transplants. And again, you can see that in African Americans, indeed, you have less 0% CPRA patients. You have more sensitized patients. But look what happens when you look at the patients that are coming for repeat transplants. And in the African American population, we're now over 60% patients that are greater than 80% CPRA, by far much more than any of the other populations. So the question is, are we actually helping or doing exactly the opposite when we're trying to avoid matching? And there are obviously a lot of facets to this, and we can have more discussions, but purely looking at generation of HLA antibodies, I think this is a very, very critical point. So we've reasoned, oops, I just missed a slide here. We've reasoned that we have this kind of vicious cycle that takes us from the first kidney to not transplanting with a matched donor, to developing de novo antibodies, to rejecting the organ and having to repeat the transplant, but now we're more sensitized, et cetera, et cetera, et cetera, and talked about all the things that this vicious cycle does at the patient level, and actually also at the societal level. And I think those are considerations that someone need to pause for a minute and think about it, and maybe try to find some ways to address them with a little bit out of the box thinking. I said that the SRTR data cannot help us resolve the role of DQ. So what I've talked about until now was just HLA mismatching at large, nothing specific for DQ. There are a couple of publications that try to look at this with inconclusive roles. We've tried to do the same, and I'm not going to show you any of the data, but really just to show that we took a similar cohort. We did not look at patients prior to 2010, because prior to that, we didn't have molecular typing. So I think the typing could have been inaccurate for a lot of those patients. Soon after, we had to put unacceptable antigens into the system. So CPRA information was more accurate, and we were able to get a little better sense of what's happening with DQ, at least with the 7 serological specificities compared to the 3 or 4 that we had prior to that time. But again, nothing very conclusive when we're looking at the same cohort. What we wanted to do using SRTR data is to look at it a little bit differently. We know we cannot take information about antibody-mediated rejection. We cannot tell that an organ failed due to antibody-mediated rejection. We don't have donor-specific antibody information in the SRTR. But if we look at patients that received the first transplant, and did not have donor-specific antibodies prior to that, and the assumption that the vast majority of the patients were transplanted across and negative cross-match, and actually you can get that information from the SRTR. So if it said positive cross-match, we excluded those patients. And then you can look at them returning to the wait list And now they need to have unacceptable antigens listed. And this is all at the serological level, so a significant limitation of this assay, of this study. But at least you can say, my donor was DQ2, I didn't have antibodies before, my patient is now back on the wait list, and now he has DQ2 as unacceptable antigen. It makes sense, right? Those are assumptions, but it makes sense that he developed this DQ2 new unacceptable antigen as a result of rejecting of the previous organ. So you can see the cohort that we had. We ended up not with too many patients, unfortunately, but really were very rigorous at what we've included in this. And you can look at the number of patients that return to the wait list with new donor-specific antibodies, or with no new donor-specific antibodies, unacceptable donor-specific antibodies, I'm going to call them, whether they received a deceased or a living donor transplant. And we use this cohort, the donor-specific unacceptable antigen cohort, as I'll refer to look at several things. Here, what we wanted to see is what is the probability of relisting with a donor-specific unacceptable antigen after the organ had failed for the individual loci. So you're looking at deceased donors and living donors, and the asterisks represent the p-value of less than 0.5. And you see that in both population, DQ, first donor DQ returning to the wait list with that DQ as unacceptable was more frequent compared to all the other loci, both for deceased donors and living donors. Again, the numbers are really low. P-values are not enormous, but I think this is the first time that we can show some specific correlation to new DQ antibodies as a result of a failed organ using SRTR data. We wanted to see what we can tell on this in the different ethnic population. I apologize for not having the Asians here. The numbers were too small, and the statistical advice that we got is don't present the data because it is meaningless. We really cannot do a good statistical analysis. But you can see that looking at all the different populations, DQ comes up as the strongest of them all. And more than this, the 2+ sign here means that we've done a comparison with how many patients were with DQ, DSA unacceptable at the end of this process among white patients and Hispanic patients compared to the African American. And again, you can see that the African Americans were significantly, had significantly more patients with DQ unacceptable reported compared to the white population and the Hispanic population. We tried to look also at what happens to CPRAs. It's a really tricky way to do this. You can see the overall kind of example, overall results. I'm sorry. I was very surprised to see similar effects between A and DQ. I suspect that A locus antigens, especially when we're talking about A2 that is so frequent in the population, kind of skew the results compared to the other loci, but you're seeing this for deceased donor and living donors. We try to cut it to different CPRA groups. Obviously, if you start with a CPRA of 80, there is that much that you can go up. So I think the meaningful results is just looking at the subgroup of patients that started with CPRA of 0%, and you see very similar picture to what we are seeing for overall of those patients. So again, indicating some role that is more specific for HLA-DQ. Okay, so I know matching is not something that we like to talk about, and you have the HLA laboratorians and then the surgeons and the UNOS and talking about equity and equality and so on and so forth. What can we do with this? And as I said, I think we need to find ways of thinking a little bit out of the box. So what I'm going to show you is not specific for DQ because, again, very difficult to get DQ data from the SRTR. This is an unpublished study. It was just submitted. again, with my colleagues in France, and I'm gonna refer to it as the matched donor potential. And what we asked was the question of, okay, let's look at, and I'm gonna talk about DR, and I'm gonna talk about serological specificities, because that's what I can get from the SRTR. What is the frequency of patients with any individual HLA-DR antigen? Super simplified, because we are talking about heterozygous individuals for the vast majority of the times, not homozygous. We don't have a lot of patients that are just DR4, for example. But again, just to show ways that we can start thinking out of the box. This is not the final solution, just the path. This is the frequency of the different alleles. So we have way more patients that are typed as DR4 compared to patients that are typed as DR10. If we look at the effective donor population that was used during the 2 years that we've looked at, you can see the distribution here, which is relatively similar. These are the same antigen specificity that we're looking here. But if we combine the two together and place focus on that particular portion of the slide, you can see that we have some patients that have more donors, relatively speaking, in the pool compared to the patients. So patients that are DR4 will have high likelihood of finding a matched donor. On the other hand, patients that are typed as DR11, we have a very low likelihood of finding a matched donor, because proportionally speaking, there are less donors that are DR11 compared to patients on the wait list. And you can see here among these particular antigens, DR18, which is way more frequent in African-American population, DR8 as well, DR12. And I think this is part of the things that are driving the inability of finding a matched donor to a particular ethnic group, not because of skin color. They're obviously the whole issue of socio-economical things that I'm not going to go to, but if we focus specifically on the genetics of things, the distribution of HLA antigens among the donor pool is different than that in the recipient pool. And if that is the case, why can't we consider something that is called the matched donor potential to give priority for patients with less common HLA antigens in the donor pool? So let's say about a donor, a patient who has DR18, and we know that he has really low likelihood of finding a matched donor. Why not give them an extra boost by giving them priority points for this, the same way that we're doing for the highly sensitized patients? So this is quickly summarizing this so I can leave time for questions. Again, working with my French colleagues, we've looked at cause-specific hazard ratio models, looking at the effect of different ethnicities when we account for the matched donor potential. So they've calculated this, adjusted for blood group and CPRA. So if you look at the baseline without any adjustments, this is what we see here in the boxes. The other is a gemisch of all different populations, and I would completely ignore it, but looking at Caucasians as the as our base comparison, and you can see what happens to the different ethnic groups. But if you adjust the model, including the matched donor potential, this is what we're going to see. So again, this is very simplistic, but I think it gives us some ways to think out of the box how we can improve both equity and HLA matching at the same time. So just want to really acknowledge a lot of my colleagues, some of the director in trainings that were involved in those studies. Some of you may remember Carly. He had spent a couple of years in my lab and was very helpful in that. My antibody buddies at Northwestern, Joe Leventhal and John Friedewald, and colleagues from around the world and some funding agencies. So I think with this, I will stop sharing and take questions.
Okay. Okay, sorry. I'm not the one who should need a glass of water now. You are. That was a whirlwind. And I think we'll open it up to questions from the group. I mean, you know, I think in the end, you kind of got to the balance and the equipoise of how to use the more specific testing to expand the donor pool and get the right organs to the highest risk patients and preserve them for the future. I guess my question would be is how are you proposing to expand this beyond that, so that it, donor allocation is a very volatile topic.
Yes, you can say that again. We actually have some more data that I figured I'm going to take them out. It's in submission. So, showing data in submission, it's always like, well, it may get rejected, right? So you shouldn't show things that are not peer reviewed. But I'm pretty excited about the data with a matched donor potential. So I figured I'm going to put it up and start getting criticism on the spot. But we have some ways to do things a little bit different in UNOS as well. There is a proposal right now to use some data from the NMDP that gives us frequency of the different alleles, and definitely we can take that specifically for DQ. We've looked at our own cohort looking at this. They also want to start finally giving some points for DP antibodies, but that's a very, very small portion of the population. DQ will affect a lot of them. So there's a way to adjust the way we calculate CPRA using allelic frequencies. We actually do have the DQA, DQB information for the donors in the system because we have those HLA attachments. It's just that the SRTR doesn't have a way to capture this. It requires money, obviously, but the data is there. You just need to find a way to mine it. So I think those two components require funding, but that's pretty easy. The more difficult thing will be to come with the matched donor potential because we are not homozygous individuals, right? That would have made things very difficult. I don't have a good answer. I'm not a statistician. I don't know to do how all those simulations. But I think it's a way to start thinking about that, right? Don't just say, oh, we cannot do HLA matching because it will disadvantage certain populations. Let's think about how we can do it without disadvantaging the population. And I think figuring some sort of an algorithm to how many more allocation points you're going to give individuals with low frequency HLA antigens in the donor population can solve it. The nitty gritty thing, right? I mean, I have the easy answer, right? This is how you should do it, but how to do it is going to be very difficult. I totally agree. I'll be very happy to see people start thinking about this. I'll try to help as much as I can, but yeah, you got me there.
No, I mean, it's a serious topic. Dave, did you have a question? I noticed you turned your camera on.
Yeah, that was great. These are things that I don't think about on a daily basis, but I learned a lot. And I think you've put together a really good argument for high resolution typing. I think, one of the questions I had is that it seems as though DQ alpha can affect antibodies that are primarily against DQ beta, right?
Well, so I'm going to stop you right there because both of them should be together. So the antibody recognizes the pair.
Right,
so
exactly.
Yes, it can affect the recognition totally.
Yeah, exactly. So has anybody taken a really deep dive to start to ask the question, because it could be that some recipients might have DQ alpha beta pairs that may make them tolerized to other alpha beta pairs. So it could be that there's a whole set of rules that we're not even aware of yet because somebody hasn't taken the time like you tried to do with the star analysis, but there's not enough information there, right? I mean, it seems as though you really need to have high resolution typing on top of a bunch of, DSA data. And maybe it's not possible at present, but man, this seems like it would be a very fundable topic.
Right. Yes, it is. And we are working on this. So we have our own internal cohort, which is not large. We have about 250 peers. There is what is called the International Histocompatibility Workshop that actually will take place in 3 weeks in the Netherlands. And one of the components there that I have the pleasure of leading is looking specifically at HLA-DQ antibodies. So I just got a dump of data for about 600 additional pairs. And these are all small numbers, but right, I mean, it's kind of now from all over the world, but we do have high resolution for everything. We do have all the antibody raw data that we are mining. We're comparing this with absorption elution studies. And we have some unique cells that we're playing with site-directed mutagenesis and so on and so forth. So yes, I think the goal will be eventually to look at permissible versus non-permissible mismatches. So we won't have to specifically have HLA matching, but we know these mismatches are gonna require less immunosuppression than those mismatches. And we know this for stem cell transplantation, for example, HLA-DQ is really important. And they have two groups of DP. They don't look at the actual specificity. They look at permissive and non-permissive mismatches, right? So that is really the goal. Is a lot of for my lab is going towards that direction. And yes, absolutely, that's where I think we should go.
Yeah, I just feel like, you know, more information is usually not worse. It's usually much better. It allows you to make more informed decisions, right? So I think this is fantastic.
Very interesting that there may be more permissive matches that allow people
Right.
not to, you know, to expand the opportunities for those individuals and to keep the very selected organs for individuals who are at highest risk. Since you brought up, I just want to ask, since you brought up stem cell and their permissibility, my understanding is this is all kidney. And have you started exploring the implications for other organs?
Right. So some of our patients are hearts and lungs, but I'm kind of putting them aside. We see the same story, right? I mean, the most frequent, the most detrimental is DQ. It probably will have some different aspects to it, maybe not so much because of the immunogenicity of the molecule, but because of the immune milieu that we're seeing for different organs. So I think when we figure it out for kidneys, we can look at the different parallels with the other organs. I just thought of something else to add to David's point. One of the sub-cohorts that we're trying to expand, and again, with the addition of data now from international colleagues, we can expand it much more, is what I like to call the 2 mismatch 1 DSA cohort. We have actually quite a lot of patients, about half of our patients will be transplanted against 2 DQ mismatches, but they will develop antibodies just to 1. So just that tells you that that one is more immunogenic than the other one, because everything, you know, all the stars align to mount an immune response, yet you still didn't develop immune response to that. And I was hoping it would be a rendezvous in the park. Obviously, it's extremely complicated topic to try and figure out what are the differences between these 2 mismatches. But if we're going to start looking at it this way, then I think we can derive a lot of really interesting data. And too soon for me to say anything about that.
And I mean, if you could improve, you know, the matching and prevent secondary transplants, then you would have a double bang for your buck, right? I mean, that would be a big deal. It would open up more organs for others. Absolutely. Because it preserves organs for some, and they're not going to reject and come back again. So
exactly, yeah. That's what I meant when I talked about societal implications when you have this whirlpool.
Yeah,
absolutely.
It's fantastic.
We probably have time for maybe one more question from anybody in the audience. I don't want to, you know.
Yeah, I have a quick question. Probably I missed most of the parts. So how reliable is using HLA-DQ for transplantation? So given, HLA is the most densely polymorphic region, and currently, we only genotype the most common one, and we don't know really much about that region. So how reliable is using this HLA as for transplant?
Right. So what we do is we perform molecular typing for all the loci. And I think we have good reagents to very equivocally determine the common and what we call the common and well-defined alleles. So even those that are at relatively low frequency, you can do next generation sequencing and resolve any potential ambiguities. And I do believe that this is where the field is going. This is where all stem cell transplant typing is happening. And more and more laboratories supporting solid organ transplants are moving into next generation sequencing. So it's a full resolution, no problem typing analysis. Does that answer your question?
Yeah, thank you.
Sure.
And maybe at some point is in terms of population difference, you mentioned variation between African American, Latino, and others. So what do you think is that something goes to evolution? Or what do you think is basic difference in terms of the frequency you see in different HLA?
Well, I mean, it's a common knowledge that the genetic makeup of an individual may affect their typing. Talking about generations ago, there was a geographical segregations of different populations around the world, and the whole topic of polymorphisms and anthropology, sorry about that, is a very vast topic with a lot of knowledge about this. So actually you can kind of dive it down to other populations. There are beautiful studies about South America tribes, for example, that have unique alleles that you don't see anywhere else. I think this whole topic will become obsolete in a generation of two, just because we have a lot of mixed marriages, and it's very difficult to determine the genetic background of an individual with, say, an African American mother and a Hispanic father, just to put it kind of together. So yeah, so I think we soon enough will go away from the ethnicity from the genetic perspective. Obviously, I'm not going to go into this hot topic about the social implications and self-definition in the US. I truly focus just on the genetic background, because this is what we can determine by DNA typing those individuals. And it looks like that in your lab, you actually do NGS type all your kidney patients and donors by high resolution typing. So chapeau to you guys. You are ahead of the game. This is a comment from your HLA lab, I'm assuming. Yep. Laura, you're
unmuted. Yes, thank you very much for spending the hour with our group. We really appreciate it. Well, thank you for allowing us to record it. I'm sure Kim will share that with you and with the CTIM group here. And with that, we'll finish. And you can stop recording.
Thank you, guys. Take care.